Okay, welcome back to the last breakout report back session for today. I had a very good time in the spatial transcriptomics. It was one of the best breakout sessions I've ever been in. And it's my pleasure to welcome Jeff here on stage to report back from that session.
Thank you. Thank you so much.
I have here.
Great.
The up and down. Right?
Thank you. Can we go ahead and
just click? Yeah. You have it.
just click? Does it go? There we go. There we go. All right. It's up there. All right. Well, first of all, welcome back, everybody. It's really my pleasure to give a summary of this breakout session. It's the first time I've ever put together a talk in 15 minutes, so we're going to see how this goes. But I want to thank my co-organizer of this section and this discussion, Sonia. I think it was a very successful breakout session. We had a lot of interaction from the audience, and I think it underscores the excitement there is in the community for this developing suite of technologies that can give us spatial information. So what I thought I'd do is just give a few slides of a brief summary of what we can get with these techniques and what they can do, and a highlight. This really complements the excellent introduction we got to this approaches that from Mats Nilsson earlier today. And then we'll dive into the questions and topics that arose in this section. And you'll let me know when I'm running. Yeah, great. So I think everybody, of course, is very well aware of the importance and strength of single-cell profiling and in its ability to catalog cells, but the necessity to provide spatial context to those measurements. And I just want to highlight with this slide that this context really comes in a couple of forms. The first, really, one that we don't talk about very often, is the fact that in many cells, RNAs are not uniformly distributed, and the intracellular organization within a cell actually is a very powerful form of post-transcriptional regulation. So in addition to having catalogs of cell types, we often would like to know how RNAs are organized within those cells in order to provide a better molecular underpinning of a wide variety of the functions of these cells. And of course, the most common spatial context that we typically think about is that when we dissociate cells from complex tissues, we lose a sense of how the tissue is organized. And this, again, comes on multiple scales. It could be on the level of individual cells or the level of multiple cells across larger tissue volumes. And this suite of spatially resolved approaches to single-cell or spatial transcriptomics aim to address, in different levels, these different contexts in which spatial information is so essential to biological questions. And these technologies can really be organized into a hierarchy. There are many ways to organize them, but this is the way we find most intuitive. So you have a suite of solid phase capture methods. These are methods like spatial transcriptomics, slide-seq, and high-density spatial transcriptomics, methods that essentially capture individual RNAs on spatially bar-coded arrays. You transfer those barcodes to those RNAs, and then you proceed with Illumina sequencing, and that allows you to reconstruct the spatial organization of that tissue using an approach that is untargeted, capable of capturing essentially anything that is expressed within the cell that can be captured on these beads. You have a different suite of technologies that are known as image-based techniques, and these can really be divided based on the method by which you create fluorescent signal. So for example, you can do that via in situ sequencing chemistries. This includes Mats Nilsson's in situ sequencing, FISSEQ, and StarMap. And you can also do this by fluorescently labeled oligonucleotides or fish-based methods. These include techniques like MERFISH, SeqFish, AwesomeFish, and a growing list of these approaches. And really, at their core, these technologies perform multiple rounds of essentially single molecule imaging, and the fluorescence pattern across those images allows you to define these different RNAs. And there are, just to underscore the excitement in this field and how there are new and creative ideas being developed all the time, there are now methods that are essentially proximity-based reconstruction, this technique, DNA microscopy, where you look at the frequency with which essentially bar-coded oligos are captured, and you can use that information, or I should say, are essentially linked together. You can use that information to computationally reconstruct spatial organization. And we talked briefly about the range of performances between these different technologies and the key features to keep in mind. Of course, multiplexing is an important feature. Many of these techniques are targeted on the scale of hundreds, though, as Matt's very nicely mentioned, many of these targeted approaches have the capacity for very high degrees of multiplexing, and recently, as many as 10,000 genes have been measured. So the distinction between targeted methods and untargeted methods is now starting to blur. Resolution is an important feature. Some techniques really have the highest possible optical resolution of about 100 nanometers. Others go up to 100 microns, and this, of course, sets the different types of questions you can answer with these techniques. Throughput is important as well, the number of cells that you can capture per measurement and the tissue area. Efficiency is something that has emerged as an important determinant of the types of questions you can ask with these techniques, and we talked quite a bit about this at different points in our discussion. Some technologies capture a small fraction of the transcriptome. Others, and particularly the fish-based methods, capture a very sizable amount of the molecules that are actually present in the sample. False positive rate is something that's not often talked about, but of course is important. And I think one of the things we highlighted quite a bit is the ease and availability of these techniques. And they vary from technologies that are almost entirely home-built microscopes and flow systems, to commercial kits now. And just to give you a little bit more of a detail of what can be done with these techniques, this is some examples of the spatial or the solid phase capture approaches, spatial transcriptomics, and then high-density spatial transcriptomics, and then slide-seq, all of the same olfactory bulb region of the brain. And what these very nicely do is they provide untargeted gene expression measurements across length scales of microns up to 100 microns scale resolution across millimeters squared or more of tissue. And they allow you, with known expression profiles, to go back and, in some cases, deconvolve the cell type distribution in these regions of tissues. And they're becoming easier to adopt now with commercial options and some great work by the inventors of these different approaches. To give you a sense of what you can do with image-based methods, this is an image from a fish-based method. This is merfish, actually. Each dot is a single RNA molecule. You can now identify somal boundaries and color these based on their identity. And the point I want to make here is that this gives you a direct measure of the gene expression profile within each individual cell, and also the capacity to measure the internal organization of the transcriptome, and those type of applications are now becoming more common. To give you a sense of the scale with which these measurements can be done, this was the single field of view that I showed you. This is a single 2 by 2 millimeter slice, and so you can really get a sense of how these measurements can not only define cell expression profiles and the distribution of cell types, but map gene expression across anatomical features in tissues. Okay, so let's get into what we actually discussed. So the first topic that we wanted to discuss, Sonia and I, is really what are the barriers that are faced in adopting these techniques? I think it's not a hard sell that there's important biology to be learned if you can measure space, but the major barrier people face is how do you get these techniques into your hands, and how can the people who have helped develop and innovate and improve these techniques help in this process? One of the points that was raised, which I think is a brilliant idea, is to help drive dissemination of these techniques through workshops and training sessions. Various techniques have had some of these workshops. Others, like the fish-based methods, are still planning these types of resources. Another important feature in adopting these techniques is the availability and expense and level of expertise that's required. So techniques that require custom microscope equipment may not be particularly difficult to build, but they require a more sizable upfront cost. That's true of solid phase capture methods, but that cost is typically hidden in the Illumina sequencer that's already available to you. And there are labs already adopting these technologies and overcoming these barriers. And really, I think one of the points that came out, perhaps not directly, but indirectly, is that cost may not really be the limiting agent for many people. It's really the concern about the expertise that's needed to build these techniques in-house and start to use them. And in this sense, I think it's obvious that commercialization is likely to play an important role. And we're starting to see this in different aspects of both the solid phase capture as well as some of the in situ sequencing approaches. One point that came up here is while we're thinking about adopting these, maybe we ought to be banking tissues already. And I think this is an important conversation to extend beyond our breakout session. And one of the most important features that came out of this is that essentially all these technologies can work with fresh frozen tissues. So that means that we don't yet need to know what technology will actually be adopted for a specific organ. We can already start using, hopefully, common protocols to bank these tissues, though I think it's definitely worth noting that there's certainly some work that needs to be done to establish how long tissues can be stored and how tissue-specific these answers are. It's also interesting, FFPE, which is increasingly common, that fresh FFPE, I think, is likely possible with many of these techniques, though it's really only been tested on a few. We talked a lot about how do you combine targeted and untargeted methods. In particular, how do you go about designing panels of genes for targeted methods that might be guarded by untargeted methods, either single-cell RNA-seq or spatial methods that are untargeted. And should we really be using these untargeted methods to drive the design of targeted panels? So this is a point that has actually been discussed already in the field, and Ed shared some of the experiences of the SpaceTX group. I think this is great progress has been made in understanding this idea, but clearly this is a question that's going to be present in this field for a long time. And what's emerged is that you can design panels with unique markers per cell type, where you essentially try to find that specific gene only expressed in that cell, or you can take more of a combinatorial approach, where individual genes are actually expressed across many different cell types, but each cell type is defined by the unique combination. These appear to work quite nicely in different simulations. But one of the points that I think is important to keep in mind, and which was raised, is that when we use untargeted approaches to design panels, we have to keep in mind that those untargeted approaches have their own caveats. For example, single-cell RNA sequencing captures a modest amount of the expressed transcriptome, let's say of order 10%. And there can actually be genes that are very informative in defining cell types, as well as telling us something about the function of the biology of those cells, that are not actually easily used with this low capture efficiency to define cell types. And so technologies, spatial technologies that actually provide very high capture efficiencies, such as the fish-based methods like MERFISH, offer the ability to accurately profile these lowly expressed genes and actually discover cell types that are defined by markers that you might not have included if you had just built your panel using sequencing-based approaches. And so I think the consensus that is emerging here is that there's a lot to be learned about how one goes about designing these panels, and it's probably worthwhile thinking about trying multiple conceptual approaches and the early phases of spatial pilots on different tissues. And another question that arose is that now that we have a suite of spatial technologies that are coming together, to what degree should we start to combine information from different techniques? For example, it was pointed out by Mats Nielsen very nicely that many genes that you see strong spatial gradients in with spatial transcriptomics techniques actually don't necessarily emerge in single-cell sequencing as clearly variable genes. And so I think, of course, it's obviously important to try and combine modalities as best as you can. An important conversation that we had, which I think was a very useful conversation that, again, should continue beyond our breakout session, is to what degree should we work to develop file standards? We're in the early stages of development of these techniques. It's exactly the time where the number and types of files can proliferate, and that can actually be an impediment to analysis. So to put it provocatively, what we need is a common standard, a FASTQ file, if you will, for these type of methods. And what we've seen is that without that, it's actually an impediment to the development, to the labs that are now trying to push themselves to the pioneering edge of developing new computational techniques. They have to sort out new standards, not just between techniques, but between versions of individual techniques. And as efforts have been put in place to compare these technologies, this lack of standards has also been a pretty serious impediment. And so I think actually it might be an important time to come together as a community, or at least a subset of the community, and now define what do these standards look like? What are the best standards? To what degree can they be generalized? And this also brought us this question of, with these type of image-based data, we can easily collect massive data sets, tens to hundreds of terabytes in raw image data, and similar scales for some of the spatially resolved solid phase capture methods. And the analyzed data are much smaller, but of course there's some resistance to just discarding these raw image-based data. And so how are we going to address this problem? Should we be storing these data sets? How do we handle this issue when we aim to disseminate these approaches? And I think the consensus is that no, we shouldn't be storing these data sets. It's just simply not sensible. And yes, we should be storing at least a subset of these data sets for the goals of developing new methods. And I think there's a strong analogy to be made with next-generation sequencing. At this point, I think we all consider base calling to be a solved problem. For these spatial RNA imaging methods, I don't think we're at that point yet. I think there's a lot of room for innovation, both in calling RNAs from these stacks of images as well as cell segmentation. And so for this reason, I think it's actually very important that we select very careful benchmarks and maintain these data sets to help drive this new set of innovative analysis approaches. Additional topics came up. Essentially, all of these techniques are basically 2D, and there's a lot of biology that's lost when you take slices. I think that's important to keep in mind. There's a desire to move these techniques into 3D, but I think this is something for basic future development. And then I just want to leave you with a few major points, unless I'm okay, I can leave you with one major point. I'll actually leave you with a few. I think it's an extremely exciting time. 1 minute, yes. It's an extremely exciting time. I feel that. I think many of the members of our breakout session feel that. I hope most of the audience feels that as well. These technologies are becoming increasingly capable of providing the type of biological insights we want, and they're becoming increasingly easy to adopt, though I would not say that the barriers are real, but I would also emphasize that I think that they are lower than they might appear. And I think everyone agrees that there's a tremendous amount of promise in coupling targeted and untargeted methods, whether these are single-cell RNA sequencing or untargeted spatial methods with targeted methods. And so I think the take-home point is I think it's time to start thinking about banking tissues. It's time to think about file standards. I'm sure many of you are already thinking about this. And it's time to think about how we start storing, visualizing, and distributing these data, because we want this in place because these technologies are really ready, or almost ready, to really be distributed and used broadly. And so it's time to make sure that these critical barriers are carefully thought about, and we have solutions in place for that. So thank you.
Thanks for taking time. Any questions to Jeff? Over there.
So even though base calling, I promise this is not about genomics, genetics that way, but base calling isn't actually solved. But using that as an analogy, as the technologies for spatial resolution gets higher and higher, the things that you can do with the data, the raw data, are going to become more and more impressive, like using wavelength interference, things like that show up on the data. So throwing away the raw data, at least as the data gets better and better and higher resolution, might not be great, because other options of analyzing that are going to come down the line are going to be losing out if you throw the data away. So I just wanted to comment on that.
No, I think it's an excellent point, and that's why we want to bring this up. I mean, you know, to put it in personal terms, as a young lab that's aiming to build these technologies in-house and use them broadly, we're really staring down the problem of petabytes. I mean, we can generate that. That's not crazy for us to think about doing. And I don't know how we afford to keep it. I don't know how we afford to disseminate it. But we feel seriously, we think seriously about the issues you raised, that there's good information in there that gets discarded. And I think the solution really is to keep benchmark data sets and discard much of it, and to recognize that as technologies improve, sometimes you just don't return to the old raw data. You collect new raw data. So
I was going to ask about the same thing. This is really a crisis waiting to hit, right? When
yes.
the data size is so big. But I mean, how close are we to saying how to encode it in a smaller format and throw away the data? Like, what is that standardized smaller format, and which information should we keep and things?
Yeah,
like that?
I mean, I think we have a good set of ideas for that now. I wouldn't say that the conversation is done, or we should consider it a solved problem. I mean, there's a sense to which the obvious things, the location and identity of RNAs out of these data sets, the boundaries of cells that are found from segment costains, those are kind of the baseline. Maybe there's more. I mean, I think that's why it's now important to have this conversation, because right now we can store these data, but that's not going to be true economically for too much longer.
If you say locations of RNAs, that means you're saying that we just trust the decoding, right?
Yeah,
but
so again,
the decoding is not that mature that we can...
No, agreed, exactly. And so that's why I say the analogy to base calling, in my mind, is that I'm sure there's improvements that can be made there, so don't misinterpret my analogy. But we don't see a lot of work still being done on that, because many people trust or feel confident with the quality we get there. I think we're still on the other side of this. I feel that we've done a pretty good job in analyzing, developing algorithms that can identify RNAs, but I think there's room for improvement, and I think that's a reason why we want to keep some of this raw data to help drive that innovative new algorithms.
Okay, one more question over there.
So coming back to this issue of resolution, I think the two-dimensional resolution is really great, and it's important to increase the resolution, but much better would be to have the three-dimensional resolution increased, because then the cells would be in the context of the environment. And I think the problem in, let's say, having goodly resoluted volume data is to have thin slice thicknesses, or let's say short distances between subsequent slices. So can you comment on how thin you are and how much information can be obtained from this?
Well, so first of all, I think you're right. There's a lot of biological questions where you really need to see 3D architecture. But I'd push back a little bit and say that there's a lot of biology you can answer from a 2D slice. And so I think there's a lot we can do without yet thinking about how we extend to the third dimension, recognizing, of course, that there are questions that we are not addressing when we don't do those type of analysis. And then I think there are relatively straightforward ways to bootstrap us into 3D from essentially serial slicing and then to various degrees reconstructing what that 3D volume is. So to get to your question, many of these techniques have slices on the scale of 10, 20 microns or so. And I think it's not particularly challenging to think about if, like you said, doing every other slice or essentially keeping these slices and trying to stitch back these 3D volumes. And there's been a lot of work on this in other fields as well. So I think that's the way in which these 2D slices and 2D techniques actually move into 3 dimensions in the early, in the next stages.
But this was part of the question, because if you have, let's say, 100 nanometer dimensions in the XY direction, but 10 micrometer thick sections, then you have basically the z-axis resolution of the whole volume data. So what you need is then thinner sections, basically.
Oh, no, I think one point to make there is that just because the section is 10 microns thick, optically you can resolve the location of that RNA in the axial direction to much better than that. It depends a little bit on the optics you use, but you can resolve the location of those RNAs to micron or better in that axial direction. And so you actually can see distributions of RNA change throughout a 10 micron thickness slice pretty readily with high numerical aperture imaging.
Okay, I think we have to cut here. Thanks, Jeff, again. Next on stage, reporting for spatial proteomics, Mike Angelo, without slides, I've heard.
Yep. Slides. I was going to say, I was very unfortunate I have to follow Jeffrey, because that was a very polished, great presentation. And so Liat's going to be coming right after me, and she also has very nice slides. So you're now in the nadir of the session. But she told me that I look like Madonna with my boom mic, so I'm feeling pretty good right now. So, sorry, I do have notes, though. They're going to care if I'm going to put my computer on top of their computer. Okay. So, in our Baron had to run out, so unfortunately you're stuck with me. We talked about spatial proteomics primarily within the context of the new antibody-based imaging techniques that have emerged, both fluorescence and mass spec-based, that Baron did a great job of reviewing. And the first thing I think was the biggest reoccurring theme was that it seems like there's a lot of new talent and new blood coming into this field that want to be able to do spatial proteomics. They would like to do antibody-targeted assays, and but they are a little they want a little bit of guidance about where to seek out, for instance, people that have experience in staining tissue, experience with tissue histology, knowing like how to QC and validate these types of antibodies and tissue sections. And I think the big thing that came up was people wanting to know a lot more about how do you select an antibody? Are there sort of centralized resources that have a list of documented, well-validated antibody clones and what the tissue preparation procedures that have been validated with those clones are? And so one of the things that we kind of all agreed on was that, and I think there's efforts underway in parallel and HubMap along something like this as well, but between HubMap and HCA is to try to form a sort of a central repository where validated antibody clones can be submitted by different labs, or can be collated from the literature that essentially says the target, the vendor, the clone, and importantly, one of the things that often isn't accompanied in a lot of the IHC literature is to also be very explicit about the tissue processing procedures that were used for epitope retrieval in the case of like formalin-fixed paraffin-embedded tissue. And so that kind of led to the next sort of big topic, which was if you have these antibodies, how do you know what a good positive-negative control is? What are the standard procedures people usually do for this? And Baron and I told, sort of trials and tribulations and horror stories about, you know, bad performing antibodies, good performing antibodies, and having to use cell lines and seeing reagents actually didn't work, and And so one of the things that we thought would be important to bring up that I think is, Baron actually had a very good, he had a good analogy, which is that we really are at the point that whether it's for this or for whether it's for any of the new emerging, you know, transcriptomic-based methods, we kind of need a NIST for biological specimens. And so, you know, and this is, and so because the problem is that we need good centralized control specimens that are made and the amount that they can be distributed broadly and that testing between batches can be performed. I mean, you know, this is like kind of a bigger operation, but that you know, we kind of both felt, and I think the group eventually kind of agreed too, that most of these high-dimensional techniques, it's going to be very difficult to have anything that's tractable over time in terms of consistency, sensitivity, and all these other things, unless we start to talk about how we can centralize standard sample generation and how we supply that to like the scientific community. And so then the question came up of, well, what if there's not an antibody for the protein you want to look at? And Baron and I both said, well, then you're out of luck. No, so what we said was, you know, he's done, they've done some work, we've done some work about incorporating targeted RNA transcript visualization using existing commercial platforms like from Advanced Cell Diagnostics. It uses branch-chain DNA amplification for a handful of targets, and how we can sort of focus and build out from there to you know, supply essentially the top, you know, 20 or 40 targets for proteins, many times cytokines, especially with immune cells, are a big one. It's because those are secreted proteins. I mean, the antibodies aren't great to begin with, but because they're secreted, it makes it even more difficult unless you're looking at the transcriptome level. So we talked about possibly working on sort of more extensible development of dual modality panels that could be used across platforms. And the other two things we thought about was that, you know, this is really kind of a good opportunity for, you know, these antibody-based approaches, which are, you know, a much more limited number of targets that can be identified, but it's very sensitive, it's got a very wide dynamic range, and sort of thinking maybe more broadly along these lines of like, how does targeted imaging methods, how can they most highly synergize with the spatial transcriptomic approaches? And in terms of using that data to really guide the high-dimensional panel development for antibody panels. And maybe part of that is if, you know, for instance, some of the great work they showed before lunch on looking at single-cell RNA-seq of the retina is that if there's targets that come out of that there's no antibodies for, well, then maybe we look at, like, once someone has done a really amazing computational analysis and they're like, these are the proteins we don't have commercial vendors for that we need to fill in the gaps to be able to identify all these different lineages, that, you know, that could also become a community effort to actually just generate antibodies for those. And so then last two things that we came, how are we on time? You okay?
Yeah, 3 minutes.
3 minutes?
Yeah.
Got
4.
it, okay. So which was, and this is one that I've got a lot of really thinly informed opinions about, which was where to sample in the tissue. So this always comes up, and and so especially like, because hardly any of these techniques are really fast enough or scalable enough to just, you know, willy-nilly scan everything we want to. So at some point, whether it's which tissue section you're going to scan or which part of a tissue section you're going to scan, some choices have to be made about like how you're going to target the analysis and why. And you know, and sort of the best thing that, you know, Baron and I suggested on that was that it, you know, in these early days when they're not sort of infinitely scalable and extensible, that we sort of rely on well-known sort of canonical multicellular structures as a starting point for deep characterization across many samples. So, you know, for instance, if it's in kidney, you'd maybe want to look at, you know, X number of glomeruli from X number of patients and so on and so forth. And then the last one, of course, is the age-old debate of 2D versus 3D. And I had to play the role of the curmudgeon of like, we don't need 3D, and then then arguments ensued, and it was fine. And so no, but I think everybody acknowledges the huge value of 3D information. I think the one thing that we had pointed out was that at least at this point. Granted, there is some work, especially at Ron Germain's lab. They've done some beautiful work on clear tissue, or they've done, 5-6 plex imaging in sort of large 3D volumes. But it's still very, very challenging to do much higher than that. And so kind of what we've suggested about that is that it doesn't have to be all of one or all of the other. I would think that most practical right now is to kind of make the volume of the data generation centered around 2D data generation, but for targeted questions where this is important, that there's no reason that you couldn't, that we couldn't have specific sort of 3D studies to go along with that. And that's about it for me. Happy to answer any questions. I answered all of them. It's amazing.
So it's best for the schedule if you have no questions.
Okay.
So we thank Jeff again. And last on stage today, we have Liat reporting back on data processing and data integration.
So I think I'm in the middle. I have minimally formatted slides. And so first I want to thank all the people that joined the spatial imaging session. I think it was a very lively and interesting session, at least for me. And I want to thank GC and Giovanni for co-leading and taking notes in this session. We started in this session, first of all, we introduced ourselves and talked a little bit about who is this community and who are the people and what are they interested in. And we found, a bunch of different answers ranging across all scales of what people really want to see in spatial imaging data. So people who are interested in cellular interactions, people who are interested in the subcellular expression, as we heard before, of either RNAs or protein, up to people who are really interested in anatomical structures in the tissue. And everybody agreed that, the big advantage of these spatial imaging methods are really that they kind of bridge all of these different length scales, and that's why they're so powerful. In terms of technical points that we discussed, we kind of divided the discussion into 5 main points. Due to time constraints, we didn't really get to talk about the last one, and mostly because sharing and storage was a very heated debate, and everybody had a lot of opinions about sharing and storage and wanting to solve that, because as we've heard before, I think this is one of the really big barriers of the field at the moment. So we talked a little bit about low-level analysis and pre-processing, some high-level analysis. How do we interact with this very complicated data? How do we share it and store it in the community and to other communities? And then how should we design experiments so that they enable this sharing and storing and different layers of interaction with the data? Oh, sorry. Okay. So just a couple of the points that were discussed about the low-level analysis. The first was that everybody agreed that experts do it best, and so probably the low-level, and I'll just say what we mean by low-level analysis. So these are things that are very closely related to the actual technology that one uses. So if it's image co-registration or dealing with background or autofluorescence or noise, so a lot of things that really, stem from the nuts and bolts of the technology. And so everybody agreed that, probably these stages, and if we use the analogy that was used before, maybe the base calling stage is probably best done in-house, and experts do it best. There was a big discussion of whether we want segmentation or do we just do these pixel-based approaches or voxel approaches, and that raised a more conceptual question of, do we really want to work at the cellular level, and especially since some of these technologies that are coming up don't really give you spatial resolution, don't really give you cellular resolution, it's lower than that. And then if we don't work at a cellular level, either because of the resolution of the technology or because we just choose to do the analysis on a pixel-based approach, then how do we integrate and then how do we communicate with other communities which are used to talking about the cell as an entity, and how do we find common languages? We talked about good tools for segmentation. We agreed that there are none. No, I'm kidding. But we talked a little bit about what different tools people are using and how this is a barrier in the field and how this needs to improve. And we talked a little bit about these deep learning approaches that are now emerging to improve segmentation. With regards to high-level analysis, so we talked about what people want to see in their data, what are the questions that people want answered by these types of data. So as I said, the range of interest is very broad, from subcellular location, context for the cell phenotypes that people are discovering through, for example, single-cell RNA sequencing, cell neighborhoods. So we had a little discussion about who is talking, which cells are talking to each other, if it's only cells that are directly adjacent, maybe cells that are a little bit further apart, microanatomical structures, and then larger anatomical structures. There was really a big discussion highlighting a very urgent need in terms of data sharing and formats. So we talked about the fact that we really need to involve additional communities in this conversation. So we all agreed that we have a lot to learn from pathologists and from the neuroscience community and people who have been doing microscopy for a long time. Because really a lot of the stuff that we've discussed, which is, how do we share these large data sets? And then once we have these data sets and we've annotated, somebody circled a region and said, these are necrotic cells, how do we then go about sharing that annotation without having each and every computational lab kind of go back to the start and build up this knowledge? So how do we exchange the raw data and the metadata and the analysis on top of it? We talked about a need to really generate a platform that integrates these three different aspects that are currently done in different platforms. So that is data storage, data visualization, and data annotation. So if there was one centralized place where you could kind of do all these three together, that would be, I think, a very useful tool for the community and also for bringing other people into the community. We talked about what do we share. So, if again we take the sequencing analogy, do we share raw reads? Do we share accounts? What do we share? And then, we talked about the fact that maybe we would like to share the data at multiple different levels. So maybe we would want to share the raw data, and then maybe the segmentation masks, and then maybe the cell annotations, and then maybe multicellular annotations, and that we should have some kind of format that allows us to share the data in all these different levels so that different people could come in and interact with the data at the level that they're interested in. So if somebody wants to develop a segmentation algorithm, they'll use the raw data. But if somebody cares about anatomical structures, they don't have to go and redo your entire segmentation, and they can just work at a higher level. There was some discussion about whether we want one format that everybody will now start adhering to, or do we want to generate platforms that are flexible and nimble enough at the moment to kind of feed in different formats. I think this was a good point that we didn't get to a decision, but definitely there were a lot of opinions. And then how do we share metadata along with all this information? And finally, about the experimental design, really, we didn't have a lot of time to discuss this. We talked about the advantage that can be gained, and people have discussed this before, by integrating these imaging approaches with single-cell data. And then, how do we now start, in addition to just having labeled cells and, counting expression, how do we now start really extracting morphological features from these cells, and then, having that as attributes that we can also share across the community, and design the experiment so that we can really extract this kind of data from the images. Yeah, I think that was basically it. It was a very interesting session. So thank you.
Any questions? Don't be shy.
Yes, Chloe.
This was a great summary, and I guess that question could have gone to any of the leaders of the different sessions, but did any group, or at least your group, discuss protein versus RNA imaging, pros and cons in terms of the type of data?
Yeah, everybody agreed protein was better.
Okay.
No, I'm kidding. No, so there was some talk about this. I mean, I think everybody agrees that you can extract different kinds of information by looking at the RNA or at the protein. There was a lot of agreement about the fact that they have different advantages and disadvantages. Protein is very targeted. You can measure less. RNA, you can measure more. On the other hand, with protein, you can really see, whether, it's beta-catenin, if it's on the membrane or if it's in the nucleus, it's doing different things, right? So you kind of link it back to the mechanism of the protein. I think that currently the stage is that people are following their heart in the sense that everybody does what they like, right? It's a very emotional field, emotional question, RNA versus protein. Don't go there, right? No, but we did talk about, in terms of, because this was a very data analysis-driven session, I mean, we talked about the fact that some algorithms really operate at the level where this is essential, so you really want to know whether your data was derived from RNA or protein, but then some of the algorithms that people are developing are actually at a higher level than that. When you talk about multicellular structures and you're defining microanatomical regions, you don't really care if the underlying measurements came from RNA or from protein, and really these approaches can then be shared across the communities. Yeah.
Thank you.
Okay. If there are no more questions, thank you again.